Hello everyone and welcome back to my channel. Today we will be going to study hybridoma technology. So it was given by Kohler and Milstein. So hybridoma technology is a technology which is used to produce monoclonal antibodies. Now what are monoclonal antibodies? So they are the antibodies which are produced against a single epitope of antigen. Here we can see that this is the antigen and these are the epitopes which are present on the antigen, and against them antibodies are produced. Now since a single antibody is produced against a single antigen and we are getting a pool of antibodies against the same epitope, it is therefore a monoclonal antibody. On the other hand, if we see here, antibodies which are produced against many epitopes of antigen, they are called polyclonal antibodies. Now here we can see that these antigens have various epitopes against which the antibodies are produced. So in this case, we are having a pool of antibodies against different epitopes of antigens, and thus it is polyclonal. Now, what is the need of this monoclonal antibodies? So they can be used for Western blot, immunofluorescence, ELISA, chromatin immunoprecipitation, and for other diagnostic purposes. Now, what is the basic idea behind this technology and how do we produce the monoclonal antibodies? So we basically have a cancer cell which is immortal, and we have a normal B cell which produces the antibody. Now, we want to make a cell which can produce antibody till the time we want. Okay, so what we do, we take the cancer cell, we take the B cell and we fuse them, and what we get is a hybrid which produces antibodies till the time we want because it has the cancerous property now. So how do we do it? So suppose we have a culture plate and we put our cancerous cells into it, we put our B cells into it, and we let them fuse. Okay, now there are five possibilities which can occur. We can have unfused B cell, we can have fused B cells, we can have fused cancerous cells, we can have fused unfused cancer cells, we can have fused cancer and B cell. Okay, so we have fused cancer and B cell which we want. Okay, so now in culture plate we have all these five possibilities. Now our aim is to screen the hybrid. How do we do that? So every cell needs DNA to divide, and DNA is made up of nucleotides. So the answer lies in the nucleotide biosynthesis. Nucleotide biosynthesis involves two pathways. One is salvage pathway and one is de novo pathway. Okay, so in salvage pathway, we have an activated ribose plus base, thymidine, hypoxanthine, and one enzyme is involved. Very important enzyme is HGPRT. So for salvage pathway, we need this HGPRT enzyme to synthesize the nucleotide. In de novo pathway, we have activated ribose plus ATP, CO2, and we produces the nucleotide. Now, for screening purposes, we will use a media known as HAT media. H is for hypoxanthine, A is for aminopterin, and T for thymidine. Okay, so we will culture these cells now in HAT media. Now, unfused B cells will die after some time because they have a limited time span. Any normal cell could survive only until 7 passages. After it, it will die. Okay, and fused B cell will also die because it has a limited time span. Now, unfused cancer cells will not die, but it will die in the HAT media because it is HGPRT negative. Now, in cancer cell, de novo pathway does not occur, and since it does not have HGPRT, so it will not survive in the HAT media. Okay. So in cancer cell, your salvage pathway cannot happen and it survive with the de novo pathway. Okay, since here it does not have HGPRT, it will die in heart media. Similarly, fused cancer cell will also die in heart media because they are HGPRT negative. And since in the hybrid, HGPRT is um, given by the B cell and the immortality is given by the cancer cell. So this will survive in the heart media, and now our antibodies are ready. So we can produce antibodies in this way. So this was all about the hybridoma technology. If you like it, please subscribe to my channel and let me know the topics you need. Thank you.